Okay, let us move on to the next speaker. Next we have Akul Gupta, who graduated from University of California, Santa Cruz, and was working in this project that he's going to talk about in Allen Institute. He's going to talk about recurrent neural network, reveal distinct signal flow in mouse visual cortex following expectation violations.
Hi, thanks for the introduction. Yeah, my name's Akul. I was an intern at Allen Institute working with Farzaneh, and we were interested in seeing how, we were interested in studying cortical column interactions in the mouse visual cortex using RNNs in response to novel images. So let me first start off with motivation. So we wanted to get a better understanding of the predictive coding framework. So quickly, the predictive processing framework speculates that the brain makes predictions about external inputs and updates these predictions when they don't match the inputs. However, it's not clear right now how different cortical areas and at different cortical depths interact with each other to represent and update these predictions. So here we wanted to leverage RNNs to get insight into the circuitry that allows predictive coding. So at the Allen Institute, we have mesoscope data. So what that is simultaneous imaging of 8 cortical planes. So we have 4 depths in 2 visual areas, V1 and LM. And so we have 3 mouse lines tagged for excitatory cells, and we have 2 inhibitory populations, SST and VIP. So what the mouse is shown is image every 500 milliseconds. So it's shown image, 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 and then randomly, there's sometimes an omission of that image when the mouse is expecting the image. So we call that an omission. And the last thing you need to know is we have 2 image sets. First is called the familiar image set. So what this is image set. These are images that the mice has seen before it's trained on these. It recognizes these images. And then on a new day, we'll switch to a different image set called the novel image set. These are images that it's never seen before, so they're novel. So the methodology for training the RNN. So the number of units in the RNN is the number of recorded neurons. So if I have 500 recorded neurons, I'll have 500 units in the RNN, and each unit in the RNN is fit to the activity of the recorded neuron. So in total, so this, what I'm showing you here is an omission aligned trace. So this is what I call one trial, and then I'll have around 200 of these trials. And so so over here on the bottom, this is the input to the RNN. This is the input trace. So this is the stimulus information, whether the image is present or not present. And in blue, this is the population average of the recorded neurons. And in orange is the average of the reconstruction of the RNN. So this is the same thing, except at the individual neuron level. So these are individual neuron traces. And you can see the RNN is doing a pretty good job of fitting the neural activity. So now I'm going to talk about the RNN weights, which is the results. And RNN weight magnitudes are informative. They tell us about the connectivity strength between different neurons. So the first thing that I'm going to focus on is what I'm showing you here are the input weights to the RNN. So the RNN has 2 sets of weights, inputs and recurrent weights. So the input weights are the weights from the stimulus information to neurons. So the first thing I'm going to focus on is excitatory weights. So we have 8 planes here. Like I said, 4 planes in LM, 4 planes in V1. And what I'm showing you here is the percent change from familiar to novel. So we're looking at what's changing from the familiar session going on to the novel session. And we can see here, the thing that strikes out is in deep LM, we see a strengthening of weights for the excitatory cell line. Moving on to SST, the inhibitory cell, we see across the board decreasing in weight strengths, specifically in deep planes of LM and deep planes of V1. So also this is coming back to biology, and we see that for the novel session, SST has smaller image responses. So this correlates well with what the RNN is finding. And also looking at VIP, we see the opposite effect where where we are getting an increase in strengths. Also, we see them particularly in deep LM and V1. So the same thing, the traces in biology, we see that VIP neurons are responsive to images in the novel session, but not responsive in the familiar session. So in the RNN, we see these change of weights reflecting what we've seen in biology. Okay, so those were the input weights. So now I'm going to be talking about recurrent weights. These are the weights going from neuron to neuron, so how different neurons are connecting with each other. And so this column is feedback weights, so from LM to V1, and then this is feed forward, V1 to LM. So first off, in excitatory neurons, we're seeing in the novel session, we're seeing increase in weights from deep LM to V1 and V1 to deep LM. So mutual connections with deep LM are increasing. For SST, the inhibitory, we're seeing across the board decrease in weights with no particular preference on any plane. However, interestingly, we see in VIP, we see an increase in weight from LM to superficial layers in V1. And so this is interesting because in literature, top-down predictory inputs have been studied to come from, to project onto superficial layers. So the takeaways here are that RNNs can reveal functional connectivity changes across cortical areas to novel images. And this is something that we can't do with correlation analysis. So this is really great. Also, I've shown you how different cell types have distinct changes in connectivity, excitatory, increase in deep layers. With VIP, the input weights are increasing across most layers, and recurrent weights, feedback from LM to superficial V1 is increasing. And with SST, we see mostly increase in deep layers for input weights and recurrent weights with no preference. So overall, response to novel images, we see increased excitatory feedback in LM and increased feedback to VIP neurons in superficial, which is important because VIP neurons have been studied and they're thought to carry the prediction signal or prediction error signal. So RNNs give us a further way to study this. So I'd just like to thank Farzaneh, my mentor. She's great help. We've had so many conversations and it's been just really fun working with her. I'd like to thank the Allen Institute for Brain Sciences and the Neuromatch Conference organizers. This is a cool conference.
I agree. Thank you very much, Akul. It was super interesting. We have one question from Cole Horwitz. It seems as though the largest mistake that the RNN makes when predicting mean population activity is around the same time as the omitted image. Do you have any intuition about why discrepancy occurs?
So the largest mistake, so over here. Yeah, I guess one thing is, that's interesting actually. This is actually just one trial. So it's actually not the case for all the other trials. So it's actually not the case. I guess I could have put multiple figures here.
Akul, do you have another figure with more than one trial?
I don't right at this moment.
Okay.
Let me see if I should.
We still have time, so no pressure. Take your time and give us figures if you have. You don't. Are you looking for other figures, Akul, or?
Oh, yeah. I can look for this, but I won't.
It's okay. Let me ask another question, and then if we have time, we'll do that. Okay?
Yeah, sounds good.
Okay. So Robert Woodry asks, are omission elicited prediction error signals stronger than novel stimulus prediction error signals for your model? or do they elicit the same strength?
Sorry, can you repeat that? The audio's actually, I think I'll
Sure.
just read the question. I don't know where they are though.
Let me read it. Our omission elicited prediction error signals stronger than novel stimulus. Wait, it disappeared. Stronger than novel stimulus prediction error signals for your model, or do they elicit the same strength?
Okay, so I think so our omission elicted signal stronger than novel. Okay, so I see the question. So basically, the cell line that has a distinct response to the omission is the VIP cell line, which is thought to carry the prediction error signal. And so for novel stimulus, the VIP cell line, it doesn't carry that signal. So the difference is when it's the familiar image set, the prediction signal is there. However, when it's the novel image set in the VIP cell line, the prediction error is not there. So once it starts learning the novel image set, then we start seeing the prediction signal. Let me show you, and let me show you that. Yeah, I can't seem to find the share screen button anymore. It's funny.
Sometimes it's hidden. It's like you have to hover the mouse above the lower part of the screen.
Yeah, it's like playing hide and seek with me right now. Where?
Try to bring your mouse to the lower end of the screen. There is an answer for the previous question from Farzana Nafaji. So I'll read it as an answer to the previous question. To answer your question, Akul is right, but more directly, his RNN was trained on a trait that included both images and omissions. He did not test the RNN on omissions only. He plans to do that though. So we don't know if in his RNN the response is more to omissions or to novel images. Do you want to add on this, Akul?
Yeah, I think that perfectly captures it. This is trained on both images and then the omission, and then more images. So I haven't tested it out where I've only trained the omission trace. So it may be that partially that to minimize the loss, that the RNN might be preferring to just respond to images and have that and take the loss on the omission part of the data trace.
Okay, and Dr. Nafaji just continued to answer. Also, regarding the previous question, can you please repeat it? I might be able to help. I couldn't hear well. Unfortunately, we cannot bring the attendees alive, so I'll have to repeat the question. Our omission elicited prediction error signals stronger than novel stimulus prediction error signals for your model, or do they elicit the same strength? Dr. Nafaji, please let me know if you heard this.
In the meantime, could I ask a question to the speaker?
Yeah.
So I was wondering, is there an effect of sort of N back? So how long ago there was the last previous emission was, and task duration, like is there a decreasing surprise effect after a number of emissions?
Yeah, so the first part of your question, where how long ago the previous emission is, that's actually something that I haven't studied or looked into. So I don't have a clear answer for you, but for a surprise effect, yeah, after for the novel image set, once the mice continues to see these images, there is then it starts getting the predictory signal. But in the inverse case, the opposite case where it keeps getting this like emission. I don't believe there is any surprise decrease in response to the emission. So this emission signal still continues to be strong.
Okay. Do we have more questions? We still have 2 minutes before the next speaker. So we have another answer from the Faji. Akul is right. We have very few emissions, but we think if we had more emissions, we would see the prediction error reduced. Okay. Thank you very much, Akul, once more.
It's a pleasure.
Thank you. And we're moving on to our next speaker. Let me introduce Harpreet Saini. Harpreet